Hi everyone, Dr. Ahles here. In this short video, we're going to walk through the steps of meiosis, making sure to compare them to the steps with the same name in the process of mitosis. Let's start with an overview of meiosis. When we look at our images here of meiosis, the first thing I want you to notice is that meiosis has the exact same phases as mitosis did. So meiosis will start with prophase, go into metaphase, anaphase, and telophase. Notice though, we see this number 1 after each of these phases. That's because when we do meiosis, we actually go through the exact same stages a second time, like we see down here at the bottom. So prophase 1 is the first round of genetic information division. Prophase II is the beginning of the second round of that division. When we're comparing meiosis 1 with the first set of those stages to meiosis 2 with the second set of those stages, I want you to underline, highlight, star the word homologous chromosomes next to meiosis 1. The goal of meiosis 1 is to divide up the homologous chromosomes that we have. It's been a minute since we talked about homologous chromosomes, so let's make sure to define those. Homologous chromosomes are the matching chromosomes where we have one from mom and one from dad. For example, let's say that these chromosomes here are chromosome number 1. Remember that humans have 23 different pairs of chromosomes. If this is chromosome number 1, I have one pair from mom and one pair from dad. Homologous chromosomes are my matching sets. In the process of meiosis 1, we go from having 2 X-shaped homologous chromosomes down to having 1 X-shaped chromosome. When we do the process of meiosis 2, let's underline, highlight, star that we're dividing the sister chromatids. Now let's go back to our image. Sister chromatids are the identical copies of each chromosome that we see making up half of these X structures. So in step 1, meiosis 1, we took this pair of X-shaped chromosomes and we divided them from each other. In meiosis 2, we're actually going to go through and split up these sister chromatids from each other. With that in mind, let's talk about each of the stages involved in meiosis. So round 1 of meiosis, meiosis 1, its big picture goal is to diversify the genetic information. We'll start with one diploid cell, so one cell that has 2 copies of every set of genetic information. We're going to end up with 2 haploid cells. Now, in this process of meiosis 1, for us to be able to divide up our genetic information in this pattern, to go from one diploid cell to 2 haploid cells, we have to go through the process of interphase. When we do the process of interphase, remember that we copy our genetic information. We make identical sister chromatids for each of our chromosomes. So we see X-shaped structures made of 2 sister chromatids. Remember that I'm going to have one chromosome number 1 with dad's information, one chromosome number 1 with mom's information. The identical copies of that on our sister chromatids. Remember, excuse me, that the name of the location where these 2 sister chromatids attach is called a centromere. And the centromere is going to be where we'll pull them apart, but not until meiosis 2. The first stage of meiosis 1 is prophase 1. And prophase 1 is a lot like prophase in mitosis, but there's a very important difference. I want you to underline, highlight, star that in prophase 1, we do what's called crossing over. Underline, highlight, star, crossing over. Crossing over is a process where my homologous chromosomes, so the ones that have the same kinds of information on them, will pair up generating a structure like we see over here. So these chromosomes are in synapsis. They're connected to each other. Then we have portions of their genetic information that will actually be exchanged. We exchange information at places called chiasmata, or you might see it called a chiasma if there's one of them. If we go to our second picture here, notice these sets of chromosomes that are overlapping with one another, and you can see a place on this red chromosome where it now has some blue information, or on this blue chromosome where it now has some red information. When my chromosomes cross over, they're swapping information with each other. They make these connection points called chiasmata to allow them to exchange their information. To make sure that this exchange is complete, that the information is fully transferred, we're going to we build these structures called tetrads. So my groups of chromosomes that are held together because they're sharing information is called a tetrad. And we're holding them together while they do this swap in prophase I. In the process of crossing over, we take information from mom and information from dad on those chromosomes, and we trade it out. So notice how my blue chromosome had copies of my genetic information all represented here with capital letters, whereas my chromosome from dad has all of these same genes represented with lowercase letters. We form a chiasma where these chromosomes are going to exchange the information, and we end up with a chromosome that has 2 capital letters and 1 lowercase letter, or 2 lowercase letters and 1 capital letter. What we make in the process of crossing over are 2 recombinant chromosomes, which are the ones here in the middle that have crossed over, that have shared their information. But we also have some that are called non-recombinant chromosomes. Non-recombinant chromosomes haven't exchanged any genetic information, meaning they match the original information exactly. So when we do crossing over, it's not that every single chromosome exchanges information, but half of those chromosomes are going to. The recombinant chromosomes have. They have a mix of different kinds of information. Once we have done this crossing over process, we transition into pro-metaphase. And if you remember from our discussion in mitosis, the big thing that happens in pro-metaphase is that the nuclear membrane starts to break down. This is going to allow my tetrads, or my groups of chromosomes that are attached to each other, to start to move around in my cell that's dividing. So in pro-metaphase I, we connect microtubules, we connect those long protein tubes, to the centromeres of each of our homologous chromosomes. Remember that the centromere is the spot in the middle of these X-shaped structures that holds the 2 sister chromatids together. In pro-metaphase, I start to connect to those centromeres so that I can start to pull on them, can line them up. So in metaphase I, we've pulled on the centromeres from both of the homologous chromosomes. They're still attached to each other. We still have the tetrads, but we're pulling on them from the 2 opposite directions, leading them to line up in the middle of our dividing cell. When we talk about the orientation of these chromosomes, so sorry guys, the orientation of the homologous chromosomes in the center of the cell, notice that my red chromosomes aren't all on one side of the cell, or my blue chromosomes aren't all on one side of the cell. This is another way that we diversify the genetic information that's going to end up in offspring. So the cells aren't dividing all maternal and paternal one direction or the other. We're mixing that information up as we're starting to pull it apart. Now here's a good time for me to take a moment to remind us about a difference between metaphase I in meiosis compared to mitosis. When we were doing the process of mitosis, In metaphase, each of my chromosomes lined up by itself in the middle of the cell. When we're doing metaphase 1, notice that my chromosomes are going to line up in their tetrads. They're going to be the groups of 2 chromosomes that are still attached to each other in the middle of the cell, in that metaphase plate. Once I have pulled those tetrads to the middle of the cell, and I've started to pull on them very strongly, we're ultimately going to break them apart. So remember, my tetrads are the 2 X's that we're sharing information with each other. By anaphase 1, I separate those 2 X's from one another, and I start to pull them to opposite sides of the cell. When we separate these tetrads from each other, we also break those connection points where they were exchanging information. So the chiasmata break apart, which is what leaves me with this chromosome with 2 different colors, or this chromosome with 2 different colors. We pull those things apart. The tetrads are no longer connected, but I do want you to notice that the sister chromatids, each of my halves of the X, are still attached to each other. I don't separate those from each other. They stay together. In the process of anaphase, what we're pulling apart then are the homologous chromosomes. I've got chromosome number 1 going this direction and chromosome number 1 going this direction. Chromosome number 2 going this direction and chromosome number 2. We're dividing homologous chromosomes, the structures that made that tetrad together. A quick reminder that when we did mitosis, we actually divided those X-shaped structures in half in anaphase. So we have double the amount of genetic information that we see here. We're pulling those homologous chromosomes apart as opposed to dividing them and sending them in different directions. Then we transition into telophase 1. And remember that telophase and cytokinesis pretty much overlap themselves, and that's true in mitosis and in meiosis. So when we do telophase 1, or when we've reached that stage, those homologous chromosomes have fully transferred to opposite ends of the dividing cell. The process of cytokinesis is what we use to divide up the cytoplasm, which is the soupy stuff and all of the organelles. Because this stage of telophase 1 and cytokinesis is so short, we don't typically fully form a new nuclear membrane around our chromosomes. The chromosomes might decondense a little bit, but not fully. This is very different when we compare to telophase in mitosis, because when we reach telophase in mitosis, that's the end of the road. No more division happening. So when we reach telophase in mitosis, we're going to build that new nuclear membrane, and our chromosomes are going to completely loosen up, allowing the cell to go on with its normal life. But we're not doing mitosis, we're doing meiosis. So reaching telophase 1 and cytokinesis means we're halfway there. We're not all the way there yet. We're going into the process of meiosis 2. When we do meiosis 2, our goal is to now generate those gametes, now generate those things that have half of the information. So we start off with 2 cells, and we end up with 4 cells in this process of mitosis. Big difference in our numbers of cells. When we do meiosis 2, the cells we're using to do it are the ones that came straight from meiosis 1. But here's a really important note. There's no interphase process between meiosis 1 and meiosis 2, so we don't have any growth phases, and most importantly, we don't have any DNA duplication stages either. So as we divide up this genetic information, we're going to end up with haploid cells that don't have full copies of the genetic information because I did not duplicate it before I started this division process. So let's dive in. Prophase 2 and prometaphase 2. We rebuild the spindle fiber proteins, which are the things that are going to pull on our genetic information. When we are in prophase 2, however, we're going to attach our genetic information. We're going to attach my centromeres to both sides of the cell. Remember when we were in prophase I that we attached each of my homologous chromosomes to opposite sides? Now we're going to split up my two sides of this chromosome. The sister chromatids are going to be pulled in different directions. So rather than dividing up the homologous chromosomes, the X's, now we're going to split from each other. Now we're going to split one X in half. We're going to pull it in two different directions. So that's anaphase 2 for you, is when we're going to pull those chromosomes apart. Before we pull them apart, we line them up, just like we did in mitosis. Notice that our metaphase plate now just has single X chromosomes. We don't have the tetrads with the sets of 2. And notice we have 2 cells going through this process of metaphase 2 and anaphase 2. So we build the metaphase plate, then we pull on those chromosomes to split the sister chromatids from each other. Each half goes a different direction from one another. But an important note about what makes these different from each other: the chromatids that we're pulling apart don't match each other identically. They're not the exact same like they were when we did the process of mitosis. We also have 2 cells doing this, hence the 2 pictures here, as opposed to just 1. When we transition into Telophase II and cytokinesis, this is the point where we're actually going to develop 4 separate cells. So we pull the genetic information to the 2 opposite sides of the dividing cells. Now we actually build a new nuclear envelope around them. Now that DNA actually decondenses all the way into chromatin, the loose, stringy form of DNA. We go through the process of cytokinesis, where we divide up the cytoplasm, and as we divide up that cytoplasm, we're generating 4 cells, each of which have half of the genetic information. Those 4 haploid cells, those 4 gametes, can be used for the process of sexual reproduction. Now that we've talked through each of the stages of meiosis, I want you to work on identifying which stage is seen in each of the images here. Here's a wink wink nudge nudge reminder. Each stage that I identify happens twice in meiosis. So we need to make sure to label, is it prophase I or is it prophase II? Is it metaphase I or metaphase II? So as you go through and identify these stages of meiosis, make sure to include the 1 or the 2. And also take a little bit of time to sort out these stages of meiosis as well. What happens first? What happens next? Make sure we're listing the stage as well as its number, 1 or 2, to help us keep straight what's happening in each part of this process of meiosis.